Hello, my name is John O'Connor and I'm a research fellow at Queen's University Belfast. Today I'll present a review of transcriptome-based cancer cell line radiosensitivity signatures. Radiation therapy is an incredibly important treatment for cancer, and around 50% of patients will receive it over the course of their treatment. Despite advances in our understanding of how genetics contributes to radiosensitivity, methods to personalize radiation therapy have so far not translated to clinical use, although several gene signatures based on transcriptomic data have been proposed. Cell radiosensitivity can be summarized by the dose-response curve when the cells are exposed to radiation. Two common metrics I'll talk about today are the survival fraction after 2 Gray, SF2, and the area under the curve, or mean inactivation dose, or MID. The models and the testing data use microarray chips for transcriptomics, similar to the one pictured, although platforms varied across publications. Two datasets I'll be referring to today are the NCI60, which has 60 cell lines and 9 tissues, and the CCLE, which has 533 cell lines and 22 tissues with radiosensitivity data. We identified 7 gene signatures for radiosensitivity from the literature, which use microarray transcriptomics. The most prominent of these is the radiosensitivity index, or the RSI, shown in the first row of the table. The majority of these studies use the NCI60 dataset and the radiosensitivity measure of SF2. The size of the signatures in terms of how many genes are in them range from 10 for the RSI to 175 for the Amundsen signature. Prediction models included linear regression, PCA, and support vector machines, and 3 papers didn't report a prediction model at all. In vitro validation of these models has shown poor results, but despite this, clinical studies have shown promising results for the ability to separate cancer outcome trajectories. Previous works have shown that random transcriptomic gene signatures are prognostic of outcome in cancer, and that they may be confounded by cellular processes such as proliferation. So the aims of this study were to assess the in vitro accuracy of radiosensitivity prediction in a much larger dataset than previously performed, and to benchmark this data by comparing to signatures derived at random and those indicative of cellular processes. We had 3 types of signature in this review. The first are the published signatures already described. The second are signatures representative of cellular processes. These came from the Reactome database, apart from the proliferation signature, which was from the Venet et al. Paper in 2011. The third set of signatures were randomly selected from genes on the chip with 500 resamples, and their size matched the 16 signatures from the first 2 groups. So to run through the methods, the expression data and radiosensitivity measurements were extracted for each cell line and signature, and a 3-fold cross-validation was performed. Models were reproduced if parameters were available, otherwise they were trained using the same modeling approach. The models were also retrained if the radiosensitivity measure was different. Where a predictive model was not reported, PCA with enough components to cover approximately 80% of the variance was used for prediction. The mean absolute error was compared to random signatures and an intercept-only model. These are the results on the CCLE dataset. On the x-axis we have the size in terms of the number of genes in the signature, and on the y-axis we have the error in predicting radiosensitivity. The blue line represents an intercept-only model, which we know is not expected to be a good model as it's equivalent to just selecting the mean. The red line is a median accuracy random signature, and the grey box is a 95% confidence interval for that signature. The black filled dots are the published models, and the unfilled dots are the cellular process signatures. We can see that the accuracy here is poor, and all signatures fall into the 95% confidence interval of the median accuracy random signature. This graph is the equivalent results for the NCI60 dataset, on which most of these models were trained. Results are similar, with models falling into the range spanning the 95% confidence interval for the median accuracy random signature. Some discussion to close out. The random signatures showed no evidence of decreased accuracy compared to the published signatures or those representing cellular processes. Larger sample sizes, multi-omics, and more sophisticated models may improve the model accuracy, and the clinical utility of the signatures compared to random genes is still unknown. In future work, we want to compare random and published signatures in clinical cohorts and include training and testing of more sophisticated phenomenological in vitro models, as well as the incorporation of mechanistic models. With that, I'd like to thank my two supervisors, Ian Overton and Stephen McMahon, as well as the Radiation Biology Group and the Data Intensive Biomedicine Group at Queen's. Thank you.